Okay, please say present or attend to your roll call. Asad Ali.
Yes, sir.
Okay, then Pradeep Singh. Pradeep Singh. Bishal. I am marking absent. Pooja.
Yes, sir.
Megha.
Yes, sir.
Rachna.
Yes, sir.
Yuraj.
Present, sir.
Garima.
Yes, sir.
Vaishnavi.
Yes, sir.
Okay, that was Vaishnavi Kardale, right?
Yes, sir.
And Vaishnavi Malviya. Vaishnavi Malviya. Rajat Mondal. Rajat Mondal. Sri Pooja.
Yes, sir.
Sri Pooja is there.
Yes, sir.
Very good. Then Isha.
Yes, sir.
Sharwani. Sharwani Pathak. Shipra. Shipra.
Present, sir. Present, sir.
Shipra. Okay. Priyadarshi.
Present, sir.
Namrata.
Yes, sir.
Shruti.
Present, sir.
Gaurav Kumar Singh.
Present, sir.
Shivani Singh.
Yes, sir.
Surikha Verma.
Yes, sir.
Good. Arjun Yadav.
Yes, sir.
Shivani Yelpure.
Yes, sir.
Debrup Sen.
Present, sir.
Have I pronounced it right? Debrup.
Yes, sir.
Okay, thank you.
Thank you, sir.
Asta.
Present, sir.
Rahul Wakchore.
So he left.
Rahul Wakchore left. Okay.
Yes, sir.
Okay, and this Gary Bishal also left.
Yes, sir.
Excuse me, sir.
Yes, beta.
So this is Vaishnavi Malviya. Sir, there was some problem in my mic, so I couldn't speak at that time. I was trying to, but I couldn't.
Okay. Vaishnavi Malviya. Okay.
Hello, sir.
Pradeep is also here. Yes.
So even
Sir.
present, her mic is not working.
Whose mic is not working?
Shravani.
Shravani Pathak, right?
Yes, sir.
Okay, Rajat. It seems Rajat is not here. Okay, that is all. Thank you, guys. So let us start out with our topic. Okay. Okay, do you see my screen now?
Yes, sir.
I hope my screen is visible. Okay. Am I audible loud and clear?
Yes, sir.
Well, now let us get started. Till our last day of discussion, we talked about all of the databases that were present on the NCBI resource as such. Okay, now we would move on to the databases that are, external to NCBI resources. Okay, so these are specific databases that were developed independently than NCBI. Okay, and our major focus of interest would be learning databases that are specialized for maintaining the data on RNA sequences as such. Okay, so we would first in this lecture we would be dealing with miRNA base, which holds the information as well as sequence and annotation on miRNA. Okay, in the next two lecture that will take place on the same day in the afternoon session, we would talk about two more databases like LNC RNA, okay, a long non-coding RNA database, as well as siRNA databases as such. Okay, so this is what I have planned for you today for entire day. Okay, uh, what I thought was that I would complete out with the theory portion of these databases first, and maybe then in the remaining few lectures we would talk about its demonstration and practical assignments. I hope it would be right for you guys. Okay, so to begin with, in this particular topic, this is what we are going to deal with. Okay, first I would teach you a little bit biology behind these microRNAs, or what we call them as miRNAs, like what are those and how were they discovered and how they are synthesized and where do they function in biology, right? And then we would move our focus on the description of databases as such. Okay, so this miRNA base, or what we call it as MirBase, well, it has been in development right from 2004. Okay, and it has got a lot more versions that were published one after another, with each version being improved from the previous one. So we would be talking about like how this miRNA database or MirBase has evolved, starting from a very precursory look back in 2004 to what kind of advances meant that these people have done to share miRNA information till the latest release of miRBase as such, or MirBase as such. Okay, so we would be talking about the very first version that was published in 2004, that was called as the microRNA registry. Then what things happened in 2006, what things happened in 2008, what were the advances that were made in the subsequent versions as such. Okay, so let us start talking about what are these miRNAs. Okay, so these miRNAs, which we give them a short form for microRNAs, are special class of non-coding RNA genes. Okay, so these miRNAs are approximately, say, some in the range of 20 to 22 nucleotides in length, and they play very important physiological roles in biology. Okay, so they exactly comes into picture when it comes to the regulation of translation, as well as they play an important role in degradation of mRNAs. So these are the exact physiological role, and after playing these physiological role, well, they participate at various stages at cellular level. Well, say for example, in C. elegans developmental model, they are known to control the development, and in fact, it was C. elegans in which these miRNAs were discovered. We are going to talk about the discovery of miRNA in a little bit, but they are known to play a cellular level role in C. elegans by controlling the developmental stages of this worm. Okay, Now in flies, they are known to regulate cell death as well as fat metabolism. In case of plants, they are known to regulate leaf development and floral patterning. Okay, so all in all, taking from these three examples, I want to just convey that these miRNA are known to play roles. Uh, they have got very uh, conserved function throughout evolution, right? As you know that C. elegans and flies are quite well distantly related to plants and of course mammals like humans. These mRNA are also known to play important role in development of human cancers. So all of these well, humans, in plants, in flies and C. elegans, well, all these model organisms are distributed very diversely in the field of evolution, right? So these are very distantly related. Plants might have evolved quite earlier as compared to humans, and yet each and every organism do contain this microRNA that gets activity at some or the other level in their physiology. That means that this functioning of mRNA of regulating the genes at translational level is very important. So far the evolution of life is conserved on evolution of life is thought on earth. Okay, so they have got very conserved function throughout the evolution. That is what I mean to say. Okay, so now let us talk about discovery of this miRNA, how this miRNA was discovered. So it happens that this miRNA were discovered in C. elegans. Back in 1993, Victor Ambrose was working in a lab and he identified a small RNA called as lin4 RNA. And what their study concluded was that this lin4 small RNA is responsible for regulating the expression of a bigger RNA called as lin14 mRNA. Okay, this regulation of expression happens in a developmental stages when the C. elegans your organism transforms from larval stage 1 to larval stage 2. So now coming back to the life cycle of this C. elegans, you must be aware that it has got various levels of transformation it has to undergo in order to complete its life cycle. Its development begins with embryo, then it forms L1 larval stage, L2 larval stage, L3 larval stage, L4 larval stage, and finally it transforms into adult C. elegans, which reproduces again to generate embryo. Okay, so the very first class of miRNA that were discovered when the C. elegans transforms itself from L1 larval stage to L2 larval stage, and during these developmental stages, this lin4 RNA was identified that somehow regulated the expression of lin14 mRNA. So by regulation, I specifically mean that the expression of this lin4 RNA causes less production of lin14 mRNA protein product. Okay, so if there is lin4 RNA expressed in this particular larval stage, it reduces the production of lin14 mRNA. That was the ultimate conclusion of the paper that was published by Victor Ambrose in 1993. You are advised to read this particular paper to know how this lin4 RNA were discovered, what kind of experimental setup was experimental setup was performed in order to come to this conclusion. Okay, later on it was established that this lin4 RNA interact with lin14 mRNA physically in process of regulation. Okay, and it was also found that these small RNAs are subset of larger set of RNAs. Okay, so this discovery of this lin 4 RNA was just a beginning. After